Hello. Today we are going to be discussing mutations in DNA. So, what is a mutation? It is the changing of the structure of a gene resulting in a variant form that may be transmitted to subsequent generations caused by the alteration of the single base units of DNA or the deletion, insertion, or rearrangement of larger sections of genes or chromosomes. We'll talk about chromosomal mutations in a different video, but today we're just going to stick with mutation in the DNA or chromatin stranded form. So there are several different kinds of mutations, and I've grouped them here into two different categories. So I've grouped them into point mutations and frameshift mutations. So point mutations are just the changing of a single nucleotide or small groups of nucleotides. I'm grouping them as just a single point. That's how I group it up. So we're going to talk about missense, nonsense, and silent mutations, which all fall under the category of point mutations, because that's where a single nucleotide changes, a single point. And then frameshift mutations that shifts the entire three nucleotide codon frame in either one direction or another, depending on whether a nucleotide is inserted or deleted from the DNA strand. A big question that's asked a lot is what causes mutations. So mutations can literally just happen. They can be random and spontaneous, just processes in your body don't always work perfectly, or really in any organism, so there's just random and spontaneous errors that happen there. But exposure to certain things called mutagens, which are just things that can cause mutations, they can make mutations happen more frequently or commonly. So if you are exposed to any kind of radiation or light of any kind, that has the potential to mutate your DNA. Exposure to any toxic chemicals, so things like pollution, or things like tobacco, burning fumes of any kind, those are all really toxic chemicals that could be very harmful and mutate your DNA. And then stress. So high levels of stress just cause essentially the cells of your body or really any organism to not function properly, and that's where random errors can happen in that process, which would cause mutations. A lot of times we think of mutations as something negative, which for the most part they are, but some mutations can be positive or even helpful for survival or some kind of advantage, but that will be talked about in a future video. Alright, so again, describing a point mutation, so looking at the GIF on the left, that would be a mutation in just like a single point or single really distinct part of the DNA. And the first few mutations we'll talk about are point mutations. So here I have developed an original or unmutated DNA strand. Okay. So the DNA strand I have as TAC, CAA, AAG, ACC, AGG, CCG, TCC, TGC, CTC, GGG, ACT. Okay. Those letters, again, in their groups of 3 nucleotides, those are codons, so I just made this table to clearly be able to see those groupings. I have also shown what the complementary nitrogen bases would be for the mRNA. So looking from DNA to mRNA, thymine would match to adenine, adenine would match to uracil in RNA because again there's no thymine in RNA, and then cytosine matches to RNA. So again going from TAC to AUG, that's how they would match up. Then I also have that same process showing from AUG to UAC. And then lastly I have the amino acid or unit of protein that would be coded for. So I have this yellow arrow on the side of this table to show that you will always use the mRNA to code for determining what the amino acid will be. So when you use a codon chart and you look at AUG, doesn't matter again whether it's the circular one or the square one, but if you look at AUG, you will see that it is methionine, or abbreviated MET. Methionine is the start codon, so that is what indicates that, hey, this is the start of the protein, so that's kind of why I have it labeled in green for go. And then there are 3 different kinds of stop codons. One of them is UGA in the mRNA code, and UGA just codes for stop, which I have in capital letters and in red. Okay. Now, looking at the statement below, the big fat cat ate the wet rat. So it's a really good analogy to think of a protein as a sentence. Okay. Every nitrogen base or nucleotide, that is like a letter, and then every codon or 3 letters is like a word. So imagine like TAC is the word the of the DNA. So then all these amino acids together again make one big sentence. So that's kind of going to be an analogy for looking at this. So here I have demonstrated an example of a missense mutation. So I have it highlighted and in red lettering now. So that T of the TAG in the DNA strand, that was mutated, which then, if that is mutated, that is going to change the mRNA sequence, which is going to change the tRNA sequence, and in this case, it is going to change what the amino acid sequence is. So a missense mutation is just where one nucleotide is changed, and it changes the amino acid. So even though the rest of that protein is exactly the same, if even one amino acid is changed, that protein could do something completely different. Okay? Looking at our sentences, the top sentence is our normal healthy protein, the big fat cat ate the wet rat, and then say a mutation happened that turned the C of cat into a B for bat. Now it says the big fat bat ate the wet rat. That's a different sentence, it's a different word, bat for cat. So that is going to be like a completely different protein that does something else. Again, showing our original unmutated strand of DNA, and then looking at a nonsense mutation. So a nonsense mutation is where we have a change of lettering, or sorry, nucleotides. So here we have ACT now in the DNA strand, which then makes the mRNA complementary be UGA, tRNA, ACU. And UGA in mRNA coding, when you put that through a codon chart, actually codes for stop. So what happens with nonsense mutations is if the stop codon is essentially read early because of the mutation, the entire rest of the protein that was supposed to be read and made doesn't get read at all. So the size and the complexity that that protein was supposed to be is limited greatly because of a nonsense mutation, which is just single nucleotide change to end up coding for a stop codon. If we look at our sentence, the big fat cat ate the wet rat, the word rat would be like our stop codon or stop sequence. And then in our mutated sentence, the big fat rat now happens, the word rat happens way earlier in the sentence, which means that the rest of that sentence wouldn't even be read. So the same, again, concept applies. Again, our original or unmutated DNA. And then this is a silent mutation. So silent mutations are where a mutation happens. So there we can see there is a G that was mutated in the DNA, which then makes an ACC in mRNA, UGG tRNA. But the thing is that the amino acid before this mutation was also 309 or THI. So a mutation happened where a nucleotide was changed, but the amino acid ended up coding for the same amino acid. So the protein ultimately was not changed. If you watched the previous video, you saw that there's multiple different codons for the same amino acid. This is kind of like a safety mechanism. So like mutations happen all the time in DNA. This kind of limits the amount of mutations that happen by having multiple codons code for the same amino acid. And as we can see, our sentences below would be unchanged because the same words are coded for. All right, insertion mutation is one of our frameshift mutations, and it is just easier to understand these kinds of mutations when we're looking at it in just the sentence form. So if our original sentence is the big fat cat ate the wet rat, an insertion mutation would be, for example, say an L or any other nitrogen base is inserted in the sentence. And in codons, they are always made of 3 nucleotides, no matter what. So B big now is fla, tka, tat, et, u, tra, and then the next starting codon will start with T and then go on and on and on, like that, until eventually a stop codon or another word, rat, came up clearly. So with an insertion mutation, the reason it's called a frame shift is because it shifts that frame of 3 over one way or another. In this case, it's shifting it over towards the right. And odds are it's going to make a protein way longer than it should be. Not always, but generally that would be the case. And you get drastically different protein A. As we can see, all of those words pretty much after the insertion of that nucleotide changed drastically as far as what the sentence says. So wherever a frameshift mutation happens, a whole bunch of amino acids are going to be messed up. Deletion mutation was the other kind of frameshift mutation. Instead of the deletion, the frame, or again that set of 3 is shifted towards the left this time. So here the A in cat was deleted, so then that means the A of the word 8 was then brought over to be part of CTA. And then we have a sentence that's all crazy messed up. It says the big fat ta, tet, hu, et, and then it would just go on and on until a stop codon is read. Same principle as an insertion mutation. Again, when that frame is messed up, every amino acid afterwards is going to be coded for something very, very different, and you end up getting a very different kind of protein. Thank you for watching. Please share and subscribe, and remember to spread